that I'll be talking about 4 different issues related to bacterial growth and division, and talk about what the puzzles are, the experimental facts and the puzzles. So we covered yesterday self-reproduction, and I argued why the very fact of self-reproduction, which is a basic thing that all cells do, is a puzzle, even for the simplest kinds of cells, namely the bacterial cells that we talked about yesterday. And we formulated that as a problem of coordinated doubling. Okay? That all chemicals in a cell, for it to self-reproduce by binary division, is to have all, to regulate or to have all the different chemicals, thousands of chemicals inside it, from birth to division, to double their populations at the same time. So that was basically the puzzle, that coordination puzzle, which in the kind of model that I mentioned, a dynamical systems type model that I mentioned, could be seen as a fine-tuning problem, fine-tuning of initial conditions, or it could be seen as a massive regulatory problem that the cell has to solve. And the problem is that the cell doesn't have that much regulatory machinery to do all this. Where does all that come from if it has to be by regulation? So that was one puzzle, for which I hope I will talk about the solution today. How do we understand that kind of phenomenon? But before that, I will talk about these 3 experimental issues as I had planned. Okay. Yeah. So first, I will talk about the exponential growth trajectory of cells, then about bacterial growth laws, and then about cell-to-cell variability, different kinds of phenomena, and we'll see what are the puzzles here first. So this is one of the slides that I had shown yesterday, arising from the experiments on single cell, bacterial single cells. Okay, so these are E. coli. E. coli has been studied in different conditions. Okay, different conditions, different media, and for a particular medium, you can study how individual cells are growing, and this is a set of pictures pertaining to one particular cell from birth to its division. Okay, one particular, and thousands of such cells have been studied. Okay, and people have tabulated their variability. What's the same? What's different about each of these cases, and what's the same about each of these cases? Okay, so for instance. And as you can see, as time progresses, the cell increases in length with a constant width. Okay? That's the E. coli rod-shaped cell, whose length and therefore whose volume increases with time from birth up to division. Okay? And because you have these trajectories, you can ask, what is the curve? What is the trajectory of cell size? We are interested in cell volume, but here, since the width is constant, the cell size is really given by the cell length. Cell length is a good surrogate for cell volume, so people have plotted this, and what they found are interesting patterns. And the pattern that I want to show you is that between birth and division, the length of the cell varies exponentially with time. Okay? It goes from by a factor of 2. Whatever it is, it goes to a factor of 2. There's a factor of 2 growth, but this factor of 2 growth is exponential. And the first people who observed that were Schechter et al. In 1962, where they again studied cell, the lengths of cells that they saw in their culture, And here is time, minutes from cell division, and on the y-axis is the logarithm of the cell length. And these are different cases, different cells. So what they saw was again a linear behavior. The log of the length is behaving linearly with time, which means that length is increasing exponentially with time. Okay, that's what they saw. And now these experiments have been done much more precisely of the kind that I showed you in the previous slide. People have, and other experiments that have been done. So I'm showing you what people have found. Here, what they've found is, so here they are looking at a cell which grows and then it divides into two cells. And these are the traces of cell length versus time. So each of these tilted red lines is a particular generation, is one generation. So the cell grows, and at this point it divides, and therefore the length, comes down. Then it expands again, divides, length comes down, expands again, comes down, expands again. And you can see the fluctuations also. In this process of growth and division, there are fluctuations, and that's another subject that we'll talk about. But if you look at any of these trajectories, okay, it is very well approximated by the exponential. Okay, so this has been found in a number of different experiments for a few different bacteria. I should mention that not all cells from birth to growth grow exponentially in size. There are some cells some species, particularly many eukaryotic species, which don't do that, okay? But bacterial cells seem to do that, okay? So many bacterial cells seem to do that. I'll just come to, just a second, I'll just state another fact and then we'll take questions. You can also study, while you are studying these cells, you can also study the populations of individual molecules inside the cell. Okay? So you can take some particular protein and tag it with a fluorescent tag, and you can observe how the fluorescence is changing with time. As the cell is growing, the fluorescence is growing, which indicates that the number of protein molecules of that particular protein are changing with time. And so you can track how the number of, the population of an individual protein is changing with time. And those are, these traces. And again, when you look at any one of those traces, it is very well approximated by an exponential growth. And the rate of exponential growth of the protein and the rate of the exponential growth of the length is the same. Okay? So that's what seems to, these are the results. Yes, question. It does not. That's right. There are fluctuations there. Yes. That's true. Yes. There are a lot of fluctuations. Yes.
But then how do we say that, okay, that it becomes, when it becomes double, it maintains, it exactly becomes like double?
So that is, if you take a deterministic model of the cell, in terms of those rate equations, capital Xi dot is equal to Fi of X, right? Now that's a deterministic description of the cell, okay? And if one wants to, that description we realize is an approximate description, is not the whole story. There are fluctuations. But we presume that that description is giving us the average behavior across the ensemble of cells. So on average, cells are doing precisely that, okay? So if you look at that, on average, if you take this long time average, then
it's double.
Huh?
There is double.
There is doubling, okay? So, and that model is capturing the model that I talked about yesterday, which we will return to, is actually capturing the average behavior. And if that's capturing the average behavior, then that's a dynamical system which is capturing the average behavior of cells. The question is that in terms of such a model, it seems that each time the cell is, it starts from a certain thing and ends up at precisely double.
Yes.
Okay, and the question then arises: what's making it do that? And when you what's keeping the averages so tightly bound to that very special curve? That's the point. Panel G.
Yeah, because what's the length units there? Like these are like really huge numbers, 20, 40. The units.
Okay, I don't know what the units are here. I'm sorry.
Because
I don't know what units it's being measured in. It could not be microns because that's too much.
Yeah.
The cells aren't that long. Okay. So I don't know. Sorry, I don't know what the units are in this. Here, of course, they're using normalized cell length, normalized with respect to birth. Okay. And that's what's happening. So, here in this curve, the units are mentioned. So you can see the cell is starting. It's about 1 micron in width, and roughly double that in length. So 2.5 is where it's starting, microns, micrometers, and it's going up. Right? That's right. This pattern, everything that I'm talking about the phase, the so-called exponential phase of the culture, where there is abundance of food, and every cell, competition effects between cells are not that crucial. So each cell is as if each cell has an abundance of nutrients, and it's doing its stuff independently. And there is a cell-to-cell variation, but that's what they're doing. Okay, right? Okay. So let's see. Yeah. Now since exponential growth has arisen in 2 different contexts in these lectures, it's very important to distinguish between those 2 different contexts. Okay. Yesterday, what I talked about was the exponential growth of the population of bacterial cells in the culture. Right? Let me show you that slide of monos. So again, to reiterate the answer to your question, everything that I'm talking about is in this phase. Okay, that's the phase where things are abundant. Here is where things start running out. The material starts running out. We are talking about this phase. And in this phase, because what is plotted on the y-axis here is log 2 of bacterial density. Bacterial density is proportional to the number of cells. So the logarithm of the number of cells is increasing linearly in time. Okay? Number of cells is increasing linearly in time. And that is easily understood if we assume that on average, every cell is taking a fixed time to divide in this regime. Okay? All cells are dividing in a fixed amount of time, tau. Okay? That's the interdivision time. Then, if you start with 10 cells, let's say here, if you have 10 cells, then in time tau, they will become 200 cells. In time 2 tau, they'll become 400 cells, etc. And so this exponential growth is understood there's no mystery about it. As long as you assume that all cells have a fixed time which they take to divide, right? Is this point clear? There's nothing mysterious about this exponential growth. If one understands that the basis for that is that the system has entered a phase, this so-called exponential phase, in which every cell is dividing in the same amount of time. Okay, that's the explanation for that. No mystery. What I'm talking about now is another exponential. This is within a single generation, a single cell, its length is increasing exponentially with time. Or inside the cell, molecules, a molecule's population is growing exponentially in time between birth and division in a single generation. Okay? So this is puzzling. It's not so obvious to understand why this should happen. I hope the distinction between the two is clear. There we were talking about population growth, population of cells. Here we are talking of cell length, cell size, and population of molecules inside the cell. Okay, so these are populations of molecules inside the cell. Now, why is this puzzling? Why is this puzzling? This is puzzling because we want to think of the cell as some kind of a dynamical system. We ultimately want to understand the cell microscopically in terms of what it contains, and we want to describe it, in some fundamental way, right? First principles way. And the language that we have in talking about any such system is the language of dynamical systems. Okay, and typically, the kinds of thing that I wrote down, that xi dot is some function of all the x's. Okay, and for each I, you write down such an equation, and the functions fi contain all the complicated interactions. That's what it is. Okay, at the deterministic level. Okay, again, this exponential growth, as you can see, there are fluctuations, but the behavior is exponential. And at the deterministic level, one is expecting the exponential growth. So why is this puzzling for any dynamical system? I've given you here 3 examples, okay, of one-dimensional dynamical systems. So the dynamical system is dx by dt is a constant r into x to the power of alpha. Okay? So, we are trying to, we want to understand exponential growth in terms of some underlying dynamical system. And here, we take 3 examples corresponding to alpha is equal to 1, alpha less than 1, and alpha greater than 1. Okay. So, the dynamical system that we have is very much more complicated. It has n functions xi, okay, and dxi by dt is some function of all the x's, okay. So you have n coupled ordinary differential equations in that system, right? Here you have a single real variable x, nothing to couple to. It has just this kind of a behavior, okay. And now you see, so let's see, the puzzle is that exponential behavior arises only in this case and not in these cases. Okay. So this case is alpha is equal to 1. So x to the power of 1. So x dot is Rx. Okay, that's your function. And, this is the kind of equation that arises in many different contexts. In radioactivity, it arises, for example, with a negative R or something, minus x dot is minus lambda x, right? N dot is minus lambda n, right? Radioactive decay and so on. And you know that this, such an equation, you can immediately solve it, okay? And you get a solution, x of t is equal to x of 0 e to the rt. Okay, that's your exponential solution, and it's fine. That's you get exponential growth. in this case if R is positive. Okay. If R is negative, you get exponential decay. But that's exponential growth is fine if alpha is equal to 1. Alpha is equal to 1 means that it's a linear system. The right hand side, the function f, if I think of this as x dot is equal to f of x, okay, the function f of x is simply r times x. It's a linear function of x. Okay. So for a linear function, for a linear dynamical system, so therefore, and such systems when alpha is equal to 1 would be called linear dynamical system. For linear dynamical system, there's no problem. You expect that there is exponential growth. The problem is that the system that is going to be describing this bacterial cell is going to be a highly non-linear system. Okay. And let's look at these two examples of non-linear cases. When alpha is less than 1, let's say alpha is half. Okay. Then you have x dot is equal to r x to the half. And you can easily integrate this differential equation and find that x of t is given by this function, where x0 is again x at time t is equal to 0. And r appears, this r constant appears here, the initial condition appears here, and time appears here. And you can see that it is not an exponential function of time, but it's time plus something squared. So it's a power law. There's some power in. Okay, there's no exponential growth. Okay, it's this kind of a behavior. Okay. This growth is slower than exponential growth. Yeah, plotted here. Okay. That happens if the system is sublinear. That is, this alpha is less than 1. But if alpha is greater than 1, let's say alpha is 2, then you can solve it again. And here is the solution. Okay. And you see, this growth is supralinear. It's even faster than exponential growth. In fact, you can see from this behavior that at a finite value of t, the denominator will become 0, which means that x of t will become infinite. Okay. So this kind of behavior, when alpha is greater than 1, has a finite time singularity. At a finite time, the solution blows up to infinity. In this exponential case, it also goes to infinity, but it goes at infinite time. Okay. Here also it goes to infinity. It goes at infinite time and slower than this. Okay. Exponential growth arises only in the very special case of a linear dynamical system. Okay. And the puzzle is this cell, the kind of dynamical system that one will typically write down, and I'll show you examples. Okay. Is going to be non-linear. And how come we are getting exponential growth? Okay, that's possible. So let's look at this. So here is a one-dimensional example. Let's look at a higher dimensional example. I hope the puzzle is clear. Is it okay? But let's just look at a higher dimensional example for perspective. Here we have n variables. Okay, xi, and xi dot is given by fi of x, but I take a linear function. Fi is linear. Okay, so fi of x is aijxj summed over j from 1 to n, where these a's, these coefficients a's are taken to be constants. These a's are constant. Okay, so x1 dot is a12 a11 x1 plus a12 x2 plus a13 x3 plus a1n xn. Okay, the coefficients of a could be whatever, some matrix. So a is basically some constant matrix, and you can write this set of equations, of course, in matrix notation. So x now is this column vector x1 to xn, a is this matrix, and x dot, which is the column vector, which is also a column vector of dx1 by dt up to dxn by dt. This column vector is given by the matrix A times this column vector. That's the same set of equations as this. Okay. So for such a system, this is also a linear system. It's an n-dimensional linear system. It's a generalization of the one-dimensional linear system. Okay. And what happens to the solution of this? Okay. It turns out that the solution of this is not exponential. It's actually a sum of different exponentials. Okay. But at long times, if you look at it asymptotically at long times, in this sum of exponentials, one of the terms dominates, and all the other terms become negligible, and this dominant term is exponential. Okay, and basically what happens is, this is something that one can study. If people want to understand this in more detail, students want to understand it, I can describe it and discuss it in more detail. You see that if you first find out the eigenvectors of A, find the eigenvectors of A, and let's for simplicity assume that A has n independent eigenvectors. Okay, right? So A times x of lambda is the eigenvector of A with eigenvalue lambda. A x lambda is equal to lambda x lambda. That's the definition of an eigenvector. Then you see what happens is that this general solution is a sum corresponding to exponential terms e to the lambda it. Okay, so there are various terms which appear like e to the lambda it. Okay, x lambda I. Okay, so this general solution x of t will be some ci, I going from 1 to n. So x lambda I is a constant vector. It's an eigenvector. It doesn't depend upon time. These eigenvectors depend only on A. They don't depend upon time. They're constant vectors. Okay. And you multiply this vector by e to the lambda it. The corresponding eigenvalue appears here, and some constant, and sum over all of them. A general solution in this case, when A has a complete set of eigenvectors, looks like this. Okay. And so now you can see that at long times, what's going to happen? At long times, the term for which lambda I is the largest is going to grow the fastest, right? So these terms are all constants. So there is e to the lambda 1t, e to the lambda 2t, e to the and so on. And let's our notation is that lambda 1 is the largest eigenvalue of A. Okay, let lambda 1 be the largest eigenvalue, largest real part. Okay, then this term is going to grow the strongest, right? This is going to grow fastest. All of them are growing exponentially, but this will grow the fastest, and it will dominate at long times. Okay, so at long times, as t goes to infinity, this behavior essentially becomes C1 e to the lambda 1t x lambda 1. Okay, all the others can be neglected. This term is by far the largest term. Okay, and now you see that in this case, therefore, while the general solution is not a single exponential, but the asymptotic solution in general is still exponential. Okay, so what is happening here? That so this is a vector, and this is also a vector. So every component x1, x2, x3, xn, all of them are growing now with the same lambda 1t, e to the lambda 1t. Okay, so you have here not at small times. I mean at arbitrary times, but at in the end you basically have an exponential solution also. So and this is a property of the linear system. It's because of this linearity that this system that we are talking about is the linear system that you're ending up with exponential growth. And when this happens, notice that if you get a behavior like this, not only are you getting exponential growth of every chemical, let's say chemicals were to grow like this. Let's say all the chemicals. So what does this mean? This means that xi of t is equal to C1 x lambda I, x lambda 1 I, because this is a vector, it has n components. So x lambda 1 I is the ith component of this, e to the lambda 1 t. Right? That's the behavior of all of them at long times. So each one of them is behaving like this. Notice that all of them are growing at the same rate. At long times, they're all growing at the same rate. Right? So if one of them doubles, the other one will also double. If all your chemicals are executing exponential growth with the same exponent, this coefficient lambda 1, with the same growth rate lambda 1, if all your coefficients are executing exponential growth with the same growth rate, then balanced growth is automatic. Right? Balanced growth is automatic. Why? Because, well, so at time t is equal to 0, you have some constant, right? So then what this will mean is this. Look at this. If this is the behavior for each one of them, then xi of t divided by xj of t is xi of lambda divided by xj of lambda, and also you can write down c. This coefficient c1 will cancel. Okay, from both. So that's what it is. Is this clear? That the ratios are independent of time. The ratios, if all the chemicals are executing exponential growth with the same rate, then their ratios have become constant in time. And what that means is that if the cell has this kind of a behavior where xi are going to be the chemicals, then at long times, the cell will settle down into a state of balanced growth. Is this point clear? Yes. So, it turns out that for us, it's not going to be a linear system. This is what would happen if the system was a linear system. What I'm trying to say is that if our underlying dynamical system was a linear system, then balanced growth would not have surprised us. Okay? Neither would exponential trajectories have surprised us. Exponential trajectories would not have surprised us. Balanced growth would not have surprised us if we had an underlying linear system with some matrix A. Okay? But I cannot tell you how to construct this A because our systems are non-linear. Okay? They are not of this form in the first place. Okay? Yes.
The rate is same for all the chemicals, but after which time scale the rates are becoming equal?
Good question. Okay. So, if I take this solution, you want to ask after what time can I neglect the other terms and have a behavior which is essentially all of them growing with the same exponential rate. What is the time, right? Now, that time is given by the reciprocal of lambda 1 minus lambda 2. Okay, that time, let me call it t1 is equal to 1 over lambda 2 minus lambda 1 minus lambda 2. Okay, why? That follows from this. Because we want to see that after how much time can we neglect the second highest term, right? If we can neglect the second highest term, then we can neglect all the other terms also, right? So let's see which is the first correction to the leading term. Okay, the first correction to the leading term will be sort of given by, so we'll write down C1 e to the lambda 1 t x lambda 1 plus C2 e to the lambda 2 t x lambda 2 plus other terms, and then the last one will be Cn e to the lambda n t x lambda n. Right? That is this whole thing. I'm just opening out this sum, right? Now, I take e to the lambda 1 t outside. Then I get C1 x lambda 1 plus C2 e to the minus of lambda 1 minus lambda 2 t x lambda 2 plus other terms like this. So since I've extracted lambda 1 t outside, I have lambda 1 minus lambda 2 inside. Here I will have in the next one lambda 1 minus lambda 3 t and so on. Okay. Is this clear? All the other terms will go like that. Now this term, at what time will this term be essentially ignorable? When this factor becomes much smaller than unity. And when will this factor become much smaller than unity? When t becomes much greater than the reciprocal of lambda 1 minus lambda 2. Is that clear? Right? So for t much greater than 1 over lambda 1 minus lambda 2, okay, can ignore the second and other terms. Okay. So, how long will the transient last? The transient will last about this long. Beyond that, it will have reached a state of balanced truth. After this much time, it will have reached a state of balanced truth. Okay. Yeah.
And the time scale, and then find the lambda 1 and lambda 2.
That's right. If you have a linear system. Yeah. Yeah. Indeed. So, but for bacteria, we don't have one. Okay. So, yeah. Okay, a good question. So, in general, an A matrix will not. Okay, but and of course, as I've said, we don't have a simple description in terms of an A matrix for real bacteria. But the physical reason why there is actually growth is autocatalysis. That the system of chemicals that we have inside a bacterial cell, is what is called an autocatalytic chemical system. And what is an autocatalytic chemical system? An autocatalytic chemical system is one broad, one, I can give you very precise definitions of what is an autocatalytic set and all that stuff. But for present purposes, it's something in which different chemicals are cooperating to increase each other's population. Okay, so they are cooperating to help each other grow. Okay, so that's why there is actually because it is an underlying autocatalytic set, there is a topological description that one can give. Okay, a property that one can state. Okay, it is that which is ultimately responsible for why it's actually growing and not the largest eigenvalue could have been negative, as you point out. So the growth rate could be negative. So the reason why it grows is because we are talking of systems which have this property of the different components helping each other grow. It's a cooperative system of chemicals. Okay. I think. I don't know the answer. Let me put it this way. Okay, all the interesting systems that we've come across that have this property have been autocatalytic, but I don't know if there is a proof that that is a necessary condition. It's certainly not a sufficient condition, because even if you have autocatalysis, there could be other dynamical things. So you might have an autocatalytic set, but there could be effects which are depleting chemicals. there is an autocatalytic set at work which is trying getting the system chemicals to grow with time, but there could be other competing set. For example, just the degradation rates. You might just have such a high level of radioactivity that in spite of it being autocatalytic, all the chemicals are decaying because of some other external effect, and so it still doesn't grow. So it's certainly not sufficient. Whether it's necessary or not, I don't know the answer. Okay. Although most interesting cases in which there is this kind of exponential growth seem to have some kind of autocatalysis in them. Yeah. Okay.
What about delta 1 and delta 2?
Yes. So then, basically you have two eigenvectors. You have a two-dimensional space, and your eigenvector could be in any of those directions. So yes, but still, you could imagine that it would end up in an exponential growth. But of course, so the time it might take to converge that. I mean, the question is, the time of convergence would then depend upon what is the difference between lambda 1 or lambda 2, which are equal, and the next one, lambda 3. So it would still converge to exponential growth in that. In that. Yes, then you consider lambda 3. Yeah. Right. Yeah. Right. The time scale will depend upon that. Yeah. Okay. And there might be certain kinds of situations which you can end up with, in which, so if you, I mean, there are situations in which A does not have a complete set of eigenvectors, and its largest eigenvalue is degenerate. Okay, in the sense that, I mean, it doesn't have, it has only one eigenvector, but there is the next eigenvalue is the same. Okay, but it doesn't have an eigenvector, and in such cases, it takes a long time to converge. Okay, because it's still okay. So it's a long time. The time scale basically is diverging. Huh? If you take n non-Hermitian, which you can very well so in this game, typically you will end up with non-Hermitian matrices. They don't have to be symmetric matrices and so on. So then that can happen. Okay. But I've given you now a kind of a motivation for why this exponential growth is a puzzle. We expect it in linear systems, but we don't expect it in the kinds of systems that we have at hand. And if as we are going to talk about this kind of dynamical system describing a cell, okay. And here is just as an illustration. I'll come back to this model in more detail if time permits later on in this lecture or in the next lecture. But just to mention it, this is an n is equal to 3 system in which the cell has been described by just 3 coarse-grained population variables. Okay, and those coarse-grained population variables are called P, T, and R. Okay, and P stands for small molecules, molecules that are precursors out of which the macromolecules inside the cell are made. So let's say it stands for all amino acids, out of which all proteins are made. Okay, so P is the pool. P is the population of all amino acids inside the cell. Okay. T stands for proteins, which are transporter proteins and other metabolic enzymes. Okay, which are converting the food molecules, and food molecules are denoted by F, where they are outside the cell. These food molecules are being brought in by the transporter molecules and being converted into the precursor molecules, the amino acids, by this pool of enzymes, which I'm calling T. So again, this is the total population of all the metabolic enzymes, so to speak, inside the cell. And this R stands for ribosomes. It's the number of ribosomes. It's the population of ribosomes inside the cell. Because ribosomes are the machines, are the key machines which are taking these amino acids one by one and making proteins out of them. Okay, so I mean that's ribosomes are the ones which are assembling the macromolecules. Okay, the protein space. Okay, so and the ribosomes are therefore also assembling themselves because ribosomes are made of proteins. They also have RNA molecules, but they also have a lot of ribosomal protein. And so the ribosomes are also making themselves because they're making ribosomal protein. So that's what is represented here. There are 3 molecules, and that's all that is used to describe the cell. Okay, it's a coarse-grained version. So X1 is P, X2 is T, X3 is R, and so you can write down a system of 3 differential equations, one for P, one for T, one for R. Okay, as describing the cell in a coarse-grained way. And this is a particular model, okay, which I will describe in more detail later. I just want to show you these equations and show you that they are nonlinear. Okay, so the concentration of P, T, and R are appearing. There's one term which is linear, but then there are these quadratic terms which are appearing. Okay, so I will justify this. I will come back and describe this to you in more detail later on in the talk if there's time, because this is a model that I intend to discuss in detail later on. But right now, all I want to show you is that the typical models that you end up with will be nonlinear. Okay, this is nonlinear because it has quadratic pieces on the right-hand side. Okay, so if you, of course, this is written in terms of concentrations. You can write it in terms of population. So what is concentration? Concentration is given by these box brackets. Okay, box bracket. The concentration is the population divided by the volume. Population divided by the volume. And you can write these equations for the populations themselves, because our variables that I had talked about in the previous slides were the populations themselves, which had to double, right? So you can write them in terms of populations, and there again you can see that there is this nonlinear behavior. Okay, we use the nonlinear forms. Okay. So the puzzle really is, how come if this is a model of a cell, how is it going to give rise to exponential growth of a cell, right? And how will this kind of a model give rise to balanced growth, exponential growth, right? That's the puzzle that we are going to be interested in. Here is another example. This was a coarse-grained version of this more detailed model of the cell, okay, where you have external food molecules, internal food molecules, other small metabolites, then analog of ATP, ADP, amino acids, nucleotides, okay? Then, various different kinds of proteins doing various different tasks, RNA polymerase, etc., various ribosomes, of course, and so on. So, it's a more detailed model. And for such a model, well, I don't know, I think this had about 30-something variables. And, the equations look like this. Okay, so each one of them has an equation, and each equation has various terms depending upon which reactions that molecule is produced in and which molecules it is consumed in, plus terms for production, negative terms for consumption. And each one of these terms is nonlinear. Here is the table of showing you, so look, this is a cubic term actually. Okay, a cubic term, quadratic terms, and so on. So, this whole set of equations has these functions fi, which are far from linear. Okay, well, that's the kind of system that one will typically have. Okay, and so basically, this is the second puzzle. I don't want to say more about it. We'll come back to its resolution later on. So that's the second puzzle. Okay. Yes. The first puzzle is what I talked about yesterday. The puzzle of balanced growth, of them doubling together. That is the first puzzle. I mean, I had a list of 4 puzzles, okay, for experimental issues to be talked about. Which okay. So the first one is that of balanced growth. Why do they double together? The second is why do they grow exponentially within a generation? Okay, so that's and the third set of things that I wanted to talk about was these bacterial growth laws. Okay, so there are certain observed regularities, which are surprising, in such a complicated system that you can have such simple empirical regularities is quite surprising. Okay, and what are they? So I'll just tell you what they are. Okay, but I don't want to spend too much time because I want to at least finish with the puzzles today. If I don't get to the resolution at all today, at least I want to finish with the puzzle. So I will state it briefly. So Mono also observed the fact that if you are in this exponential phase, so now he's looking at, in his curve, he's looking at bacteria in the exponential phase. And he is measuring the growth rate of the bacteria. But he is doing so now in different media. Okay, so he's taking the same bacterial strain, but placing it in different media. And for each medium, he will get that whole thing, right? And a part of that will be the exponential growth phase. So he measures the growth rate for that medium. Then he changes the medium by changing one component of the medium. Let's say you need a carbon source. So let's say the carbon source was glucose. In the medium, he changes the concentration of glucose. Okay, the new medium that he does it in is with a larger concentration of glucose. Okay, and everything else remaining the same. Sodium, potassium, nitrogen, phosphorus, etc. Everything else the same, but just increase the concentration of glucose. Okay. Now this is a new medium, and again he takes, he does a completely different new experiment in which he takes the overnight culture, inoculates it into this medium, and then measures the whole, those curves. First you have the lag phase, then the acceleration, then the exponential phase, and so on. And the exponential phase, he measures the growth rate. Right? That's what he's doing. Is it clear what is being done? So these are different experiments. Each dot here corresponds to a different experiment of that bacterial culture in a different medium. And he is looking at that steady state region, that exponential growth region where it has reached that steady state, so to speak. Steady in what sense? That all cells are now doubling in the same amount of time, tau. Okay? As long as that phase lasts, all the cells will double in the same time, tau. Okay, that's steady state. Okay, so that, and in that, what is the growth rate? He measures the growth rate and plots it on the y-axis, divisions per hour. Okay, divisions per hour is the rate at which the cells are dividing, right? That's just the slope of that curve, right? He's plotting the slope essentially. In the exponential phase, the divisions per hour is the slope of that curve, of that exponential curve. Is that clear? So that is what he's plotting versus the concentration of glucose in that medium. Okay. And what he finds is that as you increase the concentration of glucose, the growth rate increases. Not surprising. You made the medium richer, it'll keep, if you make the medium richer, it has more carbon, it has more nutrients essentially, and therefore it will grow faster. But this will eventually saturate. Okay. Now, after that, you can keep on adding more glucose. It will not grow any faster. Okay. That slope of that exponential part of that curve, that slope will reach the maximum possible value, and that's what we. Okay. So this is one fact. This is not surprising. Okay. This is he quantified this, and an equation like this is what sort of explains it. I mean, sort of captures it phenomenologically, that the growth depends upon the concentration. Fe is the external food source concentration. So glucose is a food source, the concentration of glucose is what enters here. And as a function of the concentration of glucose, it's given by a function like this. And you can see that this function has the property that when the concentration is small, when the concentration is much smaller than C1, for example, C1 is a constant. When the concentration is much smaller than C1, then you can, in the denominator, you can ignore this part. So it is just Fe divided by C1, and of course there's a constant here. And then basically it's linearly proportional to Fe. Right? For small glucose concentration, it's proportional to the glucose concentration. That's this part. So as the glucose concentration increases, it also increases. Okay. But once the glucose concentration goes beyond C1 and becomes much, much larger than C1, then in the denominator you can ignore C1, and then you see it is glucose concentration divided by glucose concentration, which cancels out. And so you're just left with this constant mu infinity, and it saturates. So this is a function that saturates. So that's this behavior is captured by a function like this. So this is while it is not surprising that this should happen, because after all, glucose is not the only thing that matters. There are many other things. That's right. There's nothing more. This is not surprising. I mean, this is just fitting the curve. So there's no big deal about this. Okay.
But why is that? Nature is doing that. It's not explained.
He's not explaining. So I'm giving you. I'm telling you why it's not surprising. It's not surprising because the stuff that the cell is made of doesn't contain only carbon. So glucose is providing carbon. Glucose is a hydrocarbon, so it has hydrogen, carbon, oxygen, and it is the main source of carbon. It's the only source of carbon for the cell. And all the macromolecules and the metabolites and organic molecules, they need carbon. So carbon is very important. So that's why it helps to have more carbon. Okay, but that's not what the only thing is. Not the only thing that the cell needs. Amino acids, for example, which are the building blocks of proteins. And proteins, by the way, constitute approximately more than half the dry mass of the cell. Okay? Apart from the water, most of the stuff is in proteins. That is amino acids. And amino acids require nitrogen. Okay? And so you can keep increasing the amount of carbon that the cell has. If you don't have the requisite amount of nitrogen coming into the cell, then that will become the limiting factor. Okay, so beyond a certain point, you can keep adding one nutrient, but you can't increase the growth of the cell. Okay, so the growth rate of the cell will be limited by some other nutrient. And of course, what you people find is that you can even now provide the other nutrients. Let's say you even provide the other nutrients. So then it will go to a higher growth rate. It will go to a higher growth rate. But then again, it will saturate somewhere. Okay, and eventually there are the ribosomes can make proteins only only at a certain maximum speed. So it's limited by those factors eventually. Right. So that's a explanation for it, and it's not very surprising that something like this should happen. But this curve on the right, okay, has two parts. Okay, one characterized by this equation, one characterized by this equation. And what this is doing is that it is asking, as a function of growth rate, so you are now changing the medium, and this is, let's say, some medium which is poor nutrient quality, let's say aspartate, sort of minimal medium in which the only source of carbon is aspartate. Okay, this is a better thing in which it is glucose. Okay, this is glucose. Maybe this is I don't know. So they're mentioned. Okay, and these are even richer medium in which not only you supply glucose but you supply some amino acids in the medium. Okay, so as you increase the nutrient quality, the growth rate of the cell will increase. Okay? So the x-axis is the growth rate. The y-axis has something to do with the internal composition of the cell. Okay, this is what feature of the internal composition are they talking about? essentially what they are talking about is R protein divided by total protein. Okay, they are talking about what R protein is ribosomal protein divided by total protein. Okay, this is called phi R. It's called the ribosomal protein fraction inside the cell. So, of the total amount of protein that is present inside the cell, how much of the protein is in ribosomes, is in ribosomes, and what is the rest? Okay, rest will include, for example, everything that is used in metabolism, right? Everything that is to transport chemical, food inside, to metabolize, catalyze various reactions and make the building blocks, amino acids, etc. So all those things are part of the rest. Okay. So the ribosomal protein fraction. So how much? So ribosomes are the machines which are building proteins, right? How much does the cell invest in ribosomes, in that part of the machinery of the cell, the ribosomal protein? That is plotted on the y-axis. Okay, and what is found is, so this, ribosomal protein, these proteins are, so there are so many kinds of things happening inside the cell. It's a very complicated thing. And what they find is that this is a linear function of mu. This phi r, okay, if you like, this phi r is a linear function of the growth rate. Mu is the growth rate. Okay, it says lambda here, but lambda is the same as mu. Okay, sometimes lambda is used, sometimes mu is used. Okay, so the fact that it is a simple linear function of the growth rate. So you can forget about the other details. But the fact that the ribosomal fraction is just a linear function of the growth rate is an amazing regularity that you are seeing inside the cell. Okay, so that's this equation. That's one part. That's this growth law, and this was seen by Schechter, Malo, and Kjelgaard in 1958. Okay? That's this growth law. And in 2010, Scott, this group of Terence Hua et al., they noticed this regularity, which are these curves. And what are these curves? These curves are that supposing you fix the nutrient quality. Let's say you fix, this is the case of the richest medium, this was the case of the poorest medium considered. Okay? So let's say you fix the one particular medium, any of them, and now what you do is you add an antibiotic, which influences ribosomal activity. Okay? So the antibiotic that they added was chloramphenicol. Okay? And so if you add an antibiotic which affects these ribosomes, then the proteins can't be made as conveniently as they were being made earlier, because you added an antibiotic which is doing something to the ribosomes. Okay, and so your growth rate will come down. Okay, your growth rate will come down. So, however, what they found was that the growth rate comes down for a particular medium. The growth rate comes down, but the ribosomal fraction goes up. The ribosomal fraction goes up. So you see that the same ribosomal fraction depends now negatively on the growth rate. Okay? The growth. So as you are changing this chloramphenicol level, the growth rate is decreasing, but this is increasing. Okay, that's why. And this is also linear. This behavior is also linear. Okay? So there are these constants. This phi r min is this intersection here, and this 1 over kt is the slope here. 1 over kappa t is the slope. And in these curves, phi r max is the top level, and the slopes of these curves is this 1 over kn, the negative slopes. So these are these very simple, unexpected relationships that you find between some aspects of the composition and the growth rate. And these are referred to as the bacterial growth laws of composition, because we are talking about the composition of the cell. How much of the protein is ribosomal protein? How much protein is the other protein? A fraction of what is what. Okay. How is the cell allocating its resources? Okay. So this is actually a problem that is a resource allocation problem. This is a problem of cellular economy. Okay. How does the cell allocate its resources? To ribosomes or to metabolic proteins? What are metabolic proteins doing? They are bringing in the stuff and producing the building blocks. The ribosomal proteins are taking the building blocks and making this, the proteins which are the main stuff of the cell. Okay. So where do you invest? And so that's this is a question of investment. What is the investment strategy of the cell? And this investment strategy ultimately must be contained in those functions Fi or something similar. Okay. Okay. But they found this particular property. It is called the bacterial growth laws, and we'll see how bacterial growth laws can arise from that very simple model of three pools inside the bacteria. Right? So that's okay. So this is the third property that I wanted to talk about. I've done that. And the last thing that I will talk about is the cell-to-cell variability. Okay. So again, I've run out of time, but I think I'd better finish this, okay, before we wind up today, so that on Monday I can turn to the models. So as you've seen in those curves, and it's been pointed out, that, each generation is not the same. Once the cell divides, once the cell grows and divides, the new daughters are not identical. Okay. In the next generation, when they grow, they are not exactly identical, and so there are fluctuations, and protein numbers also have fluctuations. And people have worried about this. The first, to my knowledge, people who actually quantitatively tabulated was in 1932, when they looked at cells, okay, the same cells in the same medium on different days. And on February 17th, they looked at 44 cells. On February 24th, they looked at 60 cells. And they asked the question, how long were they taking to divide? Again, they thought that they had gotten into some steady state, and they expected them to behave similarly. So they found that, some cells divided in 5 to 10, so 10 minutes. Okay, that was too fast. There was very, very little. I mean, this may have been a wrong observation. Cells actually don't divide that fast. So then they found, these cells are dividing in 15 to 20 minutes, so many, 20 to 25 minutes, so many cells, okay, and so on. So the histogram that they've plotted, cells are not taking exactly the same amount of time to divide. They are growing at different lengths of time. The interdivision time is not constant tau in the culture, but it varies from cell to cell, okay, and that's the variation. This histogram was made in 1962 by Schechter et al. Of the interdivision time and how many cells had that interdivision time. Now, these things have been measured much more precisely now. And so here is this generation time, which is the interdivision time, tau, okay, in different media, okay. And so this is the histogram. The frequency distribution or the probability distribution of how long the cells will take to divide, okay, in this medium, in which they are taking on average about 50 to 55 minutes to divide. The mean is somewhere slightly beyond 50. So the mean division time is 50. But then you can see cells which will take much longer and much shorter to divide. Okay, so that's what they see this. Sorry. Different colors correspond to different media. Okay. So this is a medium which is relatively among these medium, the poorest of the medium in terms of nutrients. So they are taking the longest to divide. And this medium is the richest medium in which they're dividing very fast. Okay, this red one. Okay? They're dividing in less than 20 minutes actually. So that's what's happening. And not only what they're doing this, you can measure this. So this is the kind of thing that is done in that experiment that I showed you with this microfluidic device and measuring cells individually. So they can also measure what is the size of the cell at division. Okay, the cell size, of course, is changing as it's growing from birth to division. So if you want to measure something, you should measure it at the same phase. So typically measurements are made either at birth or at division. Okay, so here is the distribution of division sizes. So for the richest medium, okay, the cells are the largest, and for the poorest medium, the cells are the smallest. Okay, but they all have a stable distribution. So there's a stable distribution. And so this is, and what they can do is rescale the distribution with the mean, and then they find that these distributions are almost are very similar to each other. The distributions for different media are more or less similar. Okay, they overlay on each other. So this is the division. This is the distribution of division sizes rescaled by the mean division size. Okay, SD, D stands for division. Then this is the birth size of a cell. Okay, you can also track them at birth. So this is the SB is the size of a cell at birth divided by the mean size at birth. That is this distribution. Okay, this is the generation time. And here they measured the added size. Okay, so I'll come to the I'll tell you what that is. And the funny thing that they find, so they find these distributions, and of course, it's a question: how do these distributions arise? What is the source of this variability? Can we understand them? And maybe I'll have time to talk about that. How we can. But there is something funny that they found was, okay, you see, it was expected. So it's natural to expect that, well, these distributions, let's say any one of them, let's say the division size distribution or the birth size distribution, they are these things with some variance. And the fact that, the cell basically knows that in this medium, this is my distribution. This is the distribution of birth sizes or division sizes and so on. So in every medium, the cell somehow seems to know what its size should be. Okay, and it was expected that maybe cells are making a decision to divide based on their size. See, so the question is, when does a cell divide? How does the cell know that it has to divide? Right? The cell is a bag of chemicals again. How does the cell know that it has to divide? Now it's grown big enough. Now is the time to divide, right? How does the cell know? When it's time to initiate the processes of division, which will then pinch it off and so on, or whatever it's doing. What is the division mechanism? So people thought that maybe there is the cell has a way of sensing its size, its volume. Okay. And when the volume, whenever the size goes over that threshold, then the cell basically initiates the decision to divide. Okay, initiates the division process. This is called the so-called sizer mechanism, that the cell is somehow sensing its size, and the decision to divide is based on its size. Now, if this is what the cell was doing, okay, then then you would expect. So here, what they've plotted is that they have taken on the x. Can you read, by the way, in the back or not? Can you read? No. Okay. So I'll just mention. SB is on the x-axis and SD is on the y-axis. Okay. So you take a cell, it has a birth volume, birth length, and a division length. Okay. And so you make a scatter plot. Okay. So for every cell you have a dot. Okay. Its birth size and its division size. Right. Okay. So you will get some scatter plot. You will because it's stochastic. So every cell has some birth size, some division size. You will get some scatter plot experimentally. Now supposing the division mechanism was this sizer mechanism, that the cell is sensing that now it's time to divide. I'm big enough. Okay. It's sensing effectively. It's sensing SD, right? It's sensing its size and it's dividing. In that case, whether the cell starts small or whether the cell starts big, right? It will divide at the same division length, and this scatter plot should be like this. So that if you take the average, they scatter, but so it should be horizontal. Right? You expect that? They didn't find it. Okay, they found that there is a positive correlation between the birth size and the division size. Okay, so notice the argument. Its argument is very simple. That if the cell was sensing its size and making a decision to divide based on its size, then this experiment should have yielded a scatter plot such that if you now take, if you bin it, right? If you bin these things into different bins and you take the averages across the bin, the scatter plot should have been horizontal, right? Independent of birth size, it should have, the mean division size should have been the same, right? That's not the case. Okay. So they considered another hypothesis, the timer hypothesis, not the sizer hypothesis, but the timer hypothesis. Maybe the cell knows when it was born and it's dividing after a certain amount of time has elapsed from when it was born. It's keeping track. Somehow there's something that is keeping track, a clock inside the bacterium, which is keeping track of how long it has grown, and then it is dividing whenever it has crossed, it has grown for a certain length of time. This curve disproves the timer hypothesis, because what is plotted on the x-axis is again the birth length, same SB, but what is plotted on the y-axis, so SB, and what is plotted on the y-axis is tau, the division time. Okay. And what you expect is that if it was a timer mechanism, then this plot should have been horizontal, right? The scatter plot should have been horizontal. But now you find that it's negatively correlated. Okay. That smaller cells take longer to divide. Cells which are at birth smaller take longer to divide. Larger cells divide quickly. Okay. So that's what seems to be happening. So the cells do not seem to be, E. coli cells seem to be sensing neither their size nor their age. Okay. And they're doing something else. And what they observed was the following. That if you look at, okay, sorry, not this one. Yeah, this here. Okay. If you look at the added size, if you look at SD minus SB delta. So for every cell, you can measure SB, you can measure SD. So you also have a number for for delta. How much size did the cell add? Okay. How much volume or length being proportional to volume in this case because it's growing a constant bit. How much volume has it added between birth and division? If you plot that as a scatter plot, if you make a plot of delta versus SB, and these are again plots for different media, those are horizontal. Okay. So somehow the cell seems to be sensing how much volume it has added since birth. It doesn't care what its volume is. It doesn't care how old it is since birth. It just cares about how much it has grown since birth, the added volume. Okay. And that's what it is using to divide. Okay. So these single cell experiments are telling you something about what cells are doing. Okay. And the surprises that you have. Okay. And this delta distribution then again is sort of universal in a certain sense. Okay, which we can talk about later. I think I've exceeded my time, so I will stop here. And oh yes, I had to talk about protein number fluctuations. Yeah, so this is this is the fact that it is not a sizer, not a timer, and that it is an adder. Okay, so that's the kind of yeah. Yes. True.
The bigger size, like the bacteria in the rich media and the bacteria in the poor media, the constituents inside the cell, are they, does they have any relation? Are they like the constituents are same?
That is the content of the growth laws. The growth law is telling you that as you go from rich media to poor media to rich media, how the composition inside changes. It's telling you one aspect of the composition, how the ribosomal protein fraction is changing. So the ribosomal protein fraction is changing as you go from one media to another medium. Okay, the composition is changing. Okay, and the added length is not the same. The added length is different. But the universal thing is that doesn't matter which media, E. coli is worrying about how much it has added. And for every medium, it has decided that it should add this much. Yes. So this suggests, this horizontal curves suggest that for every medium, there is a medium dependent threshold that the bacterium has for how much size it should add. Okay? And how will this arise is the question, right? These are questions. So these are now puzzles for our understanding of the bacteria. What's going on inside that explains these properties? Okay, that's the question that I will be interested in the rest of my lectures. And yeah, so that's it. There was another thing but maybe I'll skip that because in the interest of time. The protein numbers inside have very characteristic fluctuations. Okay. And they have been measured, so that's another puzzle. How do they arise? Okay. But anyway, let me not get into that. I think I'll stop here. And on Monday, then I will turn to models, which will try to explain these puzzles. Okay, and so that gives us an insight as to what is exactly happening inside the cell now. Okay.